All right, we just talked about how anatomy and physiology work together, how anatomy itself can be either gross when talking about the entire body or microscopic. Well, chemicals make up everything associated with the human body. Remember, that is at the lowest or most specific part of that ladder, that organizational ladder. All right, some basic chemistry, some important terms you need to understand. So take some notes on this and utilize maybe flashcards to make sure you understand what each of these things mean. Matter is anything that takes up space and has a measurable mass. Mass itself is that measurable amount of matter for that object. The measurable amount of matter for that object. It's usually measured in kilograms. Weight itself is the pull by gravity of an object. And then the last one has to do with elements and atoms. An element is the simplest form of matter made up of one kind of atom. An atom is the smallest component of an element that gives the chemical component to that element. Okay, you should remember basic atom structure, but let me just make sure you have it. Atoms are made up of 3 subatomic particles. You have neutrons found in the nucleus. They're neutral, so they have no charge. There's protons also found in the nucleus. Think pro, it's positive. They have a positive charge. Electrons are found outside of the nucleus in things called shells that go around the nucleus, and they have a negative charge. And some simple math associated with it. Okay, atomic number equals the number of protons. Atomic mass is the number of protons plus the number of neutrons. Okay, you can find, if we take those two things, atomic number and atomic mass, if I wanted to know what the neutron number was, all I would do is take the atomic mass and subtract the atomic number, because it's protons plus neutrons minus protons gives you neutrons. Lastly, an isotope. That is where 2 or more forms of the same element, like carbon, they have the same number of protons and neutrons, therefore the nucleus stays the same. The only thing that's different is the number of electrons, like carbon-14. So chemical bonds are formed when electrons in the outermost shell, called the valence shell, that can be shared or transferred to another atom. The type of bond is determined by their ability to attract electrons, called electronegativity. And there's a couple different types of chemical bonds. The first one's ionic, and if you look at it, ion means charged particles. So ionic, they either gain or lose an electron, therefore changing the charge. Electrons are transferred, giving a charge. So an electron is a negative charge. You imagine if I lose an electron when you're neutral, then you'd have a more positive charge. If it's neutral, let's just say it's no charge at all, and I gain an electron, you could see it would make it a negative charge. So a cation is a positive charge, and an anion is a negative charge. Now, if they're with these charges, it's like a magnet. The opposite charges attract one another. So think about on a magnet. You have 2 magnets. You have plus signs and negative signs. If you put the 2 plus signs together, what happens? It repels. But if I switch it and I have a positive next to a negative, it pulls together. And that's the idea with ionic bonds and that oppositely charged being attracted to one another. The second type of bond is a covalent bond. That's where electrons are shared. And of course, there's more than one type of covalent bond. The first one is a single covalent bond. That's where two atoms share two electrons. A double covalent bond is just double of a single. It's two atoms sharing 4 electrons. And the other two is going to be nonpolar and polar. Now remember that when you're sharing something, they either can share it equally, meaning the pull by both atoms is the same or equal. That would be nonpolar covalent. Nonpolar covalent means the electrons are shared equally and both atoms are pulling at the same amount. Polar covalent means the electrons are not being shared equally. One of the nucleus has a stronger attraction or pulling stronger than the other one. And last bond is a hydrogen bond, which is the weakest of all the bonds. It occurs when a positive charge hydrogen of one molecule is attracted to a negative charge of another. For example, water. The difference between molecules and compounds, pretty simple. Molecules are 2 or more atoms that can chemically combine to form something new. So if we have hydrogen, 2 hydrogens together would be an H2. That's a molecule. A compound is when 2 or more different types chemically combine. For example, water, H2O. H and O are completely different as opposed to the molecule H2. The molecular mass is where you add up the atomic masses for each of the portions of that total combination. So for example, water, H2O, I would take the mass of hydrogen twice. So let's just say it's 1. So 1 plus 1. And then oxygen, and take the weight of that and add it to the 2. Solubility and dissociation, I put this here because they're used interchangeably in it, and they shouldn't. It's very incorrect. Solubility means one substance can dissolve in another. One substance can dissolve by another. And like salt, if you put salt in water, it's going to be soluble. Dissociation means to separate. So compounds that are ionic will be pulled apart and bound to something else. This occurs in water. Think about it. If you're putting something in water, well, if it's a positive ion, it's going to be attracted to the negative portion of water and vice versa. That's dissociation. That's separation due to charge attraction. Okay, some chemical reactions and the energy associated with it. So a chemical reaction is when bonds are either formed or broken. And let's just look at this. So if you have a typical equation where you're mixing two things and you get a reaction, or you get something is made afterwards. So what is being put together in the reaction is called a reactant. The substance that is the result of the reaction is called the product. So this can happen whether they're put together or pulled apart. And there's some different types. There's a synthesis reaction, which I put really nice example of it. If you take A and you add B, you get AB. That's a synthesis. You're combining. Decomposition is the opposite. You take that AB and it separates into its parts, A and B. So synthesis is putting together, decomposition is breaking apart. And then there's a reversible reaction. Those are chemical reactions in which the reaction can proceed either from reactant to product or in the opposite direction, from product to reactant. Oxidation reduction reactions. Oxidation is the loss of an electron by a substance, and then reduction is the gain of an electron by a substance. Oxidation reduction reactions means it has to happen at the same time. What I mean by that is if one loses an electron at the same time, there has to be a gain of that electron by another. So if I lose an electron at the same time, you would gain that electron. That's an oxidation reduction reaction. Energy is the capacity to do work. I mean, the biggest thing to think about is you didn't sleep all night, you didn't eat well, you're exhausted. Well, your energy or your capability to do things are at its lowest. And obviously, when you're dead, you have no energy to do anything. So energy is that ability to do work, the ability to do things. And there's different types, and it's pretty cool. I've got a good example for these. Potential energy is energy that's stored. Kinetic energy is energy of motion. Think about it. You're a child and you're on a swing set. And I stand behind you and I pull that swing set up at its highest point. That's potential energy, because if I let go, something's going to happen. So holding you at that highest portion, that's potential energy. I let you go and you go forward and swing, that's kinetic energy. Mechanical energy is the energy because of motion. Chemical energy is energy that's stored in chemical bonds. So you break the bond and release the energy. And then heat energy is the transfer of energy between different temperature objects. For example, you go out early in the morning in the summer, let's just say it's 60, you touch a rock, it's cool, it's easy to grab. Well, let's take Mississippi, normal August of 95 degrees for 7 hours a day. And you grab that same rock. That rock is now hot to the touch. That's heat energy. It was being transferred. Activation energy. Now this is important when we talk about chemical reactions within the body. Activation energy is basically the minimum energy required to start a chemical reaction. The bare minimum. A catalyst is a substance that increases the rate of chemical reactions. So you might have a chemical reaction that might take 10 minutes. But if we add a catalyst to it, it could happen in a second. We'll speed up the reaction. And some examples of catalysts would be like an enzyme. Temperature, think about it, you want to make spaghetti tonight for dinner. You put just spaghetti in a pan of water. Ooh, you're going to be waiting all day and it's not going to taste the way you want to taste. What if we take that same spaghetti and put it in boiling water? Well, now I've increased the reaction rate. And then, of course, concentration of reactants. If we adjust the concentration, we can cause the reaction to speed up. And then basically just the difference between inorganic and organic. Inorganic is substances without carbon-hydrogen bonds, like oxygen. Organic then is obviously the opposite, and that is carbon-containing substances. And we'll talk about them shortly, the organics shortly. Just brief, water. Water is really important because of its unique characteristics. It's polar, which means there's different charges in the entire molecule. It can influence interaction. It can cause things to dissociate. It can cause things to bind with them. It's property where you can take water, and if you alter the temperature, you can change what it physically looks like. There is a hydrophilic end and a hydrophobic end that is involved with water. So you can either love water and be on the outside facing the water, or fear water and you'd be on the inside. Think of that plasma, the phospholipid found in a cell membrane. That hydrophilic end, it faces outward, it likes water. Hydrophobic tails are on the inside and not touching water. Water is found everywhere in the body. It has a lot of very important functions like stabilizing body temperature, lubrication of joints, or even organs within the body, maintaining a frictionless environment. It protects. I mean, if you have water in there, it's like a water balloon. I mean, it protects vital organs. It's also a medium for chemical reactions. And just think about it. You're in a bathtub, no water, and I throw in a hairdryer that's turned on and plugged in. Well, nothing's going to happen to you. What happens if you have now a bath with water in and I do the same thing? You would have an electrical shock. A couple more differences: solvent versus solute. So if we have that water and salt in the picture, like you can see, the solvent is what is being used to dissolve, so water. Solute is what is being dissolved, the salt. And then a mixture is just substances physically mixed but not chemically. It's like taking two different color sand. You mix them together and you've got brown and red sand, but there's no chemical change. Acid base, again, this is just making sure you got it. So the pH scale runs from basic at 14 to neutral at 7 to acidic all the way to 0. Your body's neutral pH level is between 7.35 and 7.45. That's where it likes to be. To say something is acidic is where it is below 7 on the pH scale, and the lower the number, the more acidic it is. For base, it means it's above that neutral number of 7, and the higher the number, the more basic it is. And then a buffer is utilized in the body, which we'll talk about as we go further on. A buffer is utilized to neutralize the acid and bring it more to a 7, a neutral level. Think about it. You have terrible heartburn. What do? You go take a Rolaids or a Tums. That is a buffer. It will neutralize the acid in your stomach. So organic chemistry, again, I'm just going over the basics. We'll go over it in more detail when we get into digestion and nutrition, but just the basics. 4 main groups. The first is carbohydrates. You have monosaccharides. Mono means one. Monosaccharides, which are your simple sugars. An example is glucose. Disaccharide, di means 2. Disaccharide is 2 simple sugars put together by a dehydration reaction, pulling out water. An example is sucrose. And the third one's polysaccharide. Poly means many. Polysaccharides are long chains of monosaccharides. For example, starch. Very difficult to break. Lipids are the second group. We're talking about fats ingested and broken down, by hydrolysis. Triglycerides are basically glycerol and fatty acids. Fatty acids could be saturated or unsaturated. Saturated means that it is made up of single bonds only in a carbon chain. So if we do a carbon chain would be like this. Here's a single bond. Here's a single bond. That's a carbon chain. That would be saturated. Unsaturated contains double bonds. So these are all single bonds. That's what that single hash means. But if I do this, That means it's a double bond in there. That would be unsaturated. Phospholipids, remember we talked about that with the cell membrane. That's where you have the polar, hydrophilic head, and non-polar, hydrophobic tails. The last thing we would mention is steroids, made up of cholesterol, and they're a ring-like structure of carbon. So if we took this carbon chain like I did, but put it into a circular shape, that would be a steroid. Third organic group would be proteins. That's where amino acids are bound together by covalent bonds. Proteins have 4 structures. You have the primary structure is just the amino acid sequence. Amino acid attached to amino acid attached to amino acid. Secondary structure is the hydrogen bond involved with that amino acid sequence actually gives it a pleat-like shape. So if you think about blinds where you pull on the string, it kind of goes up and it looks like an accordion, that's a pleating structure. Tertiary structure is a 3-dimensional structure, meaning it's not flat and one-dimensional, it's 3-dimensional, so it has you can see all around. It's dimensional. And quaternary is the last structure. That is where there is an association of 2 or more proteins together. Nucleic acids, we're talking about DNA and RNA. They're made up of nucleotides. A nucleotide is made up of 5-carbon sugar. So a 5-carbon ring, which you can see here. 1, 2, 3, 4, 5. A nitrogenous base, which we'll talk about in a second, and a phosphate. And you can see in that bottom, there it is on the bottom with the phosphate, the 5-carbon sugar, and your nitrogenous base. When we talk about DNA, this is where our genetic material is stored, and it can be passed through generations. It can be passed throughout your body when you're transcribing information for change. You're trying to make a new protein. Basically, it's 2 strands that are come together in this helix form, this twisted staircase, if you will. It is held together by specific complementary pairs, which you can kind of see on there. So you have thymine and adenine, that's the green and blue. You have cytosine and guanine, that is the orange and purple. They always together, A to T or T to A, G to C or C to G. If we separate those strands, then if I had a guanine here, I know on the other side or what should bind to it would be a cytosine. They go together. And the last thing is we're going to talk about it a billion times is energy needed. The energy that the body requires to do work that's really important is going to be ATP, adenosine triphosphate. It's the energy that is needed for chemical reaction in the body. In fact, it's just not needed, it's required. And remember, those powerhouses of the cell, those mitochondria, those produce ATP. Again, this is just a basic outline. As we go through the chapters, starting with chapter 4, we're really going to be utilizing this information much more in depth.